I'm so thankful to have you all participating in this lecture, especially to our esteemed guest lecturer, Professor Chrition and Dr. Florence from Poitiers University. We also want to apologize because the dean, Professor Prof, who's supposed to welcome you here, has another academic duty of visiting some university in UK for collaboration. However, he had conveyed his warm welcome and greeting to both of you. Professor Chrition and Dr. Florence, the School of Postgraduate Studies currently has 5 study programs, including 4 master programs and 1 doctoral degree program. The master programs are Master of Epidemiology, Master of Information System, Master of Energy, and Master of Environmental Studies, and 1 doctoral program in Environmental Studies. We are certainly still expecting that new programs will be established in the near future. Ladies and gentlemen, today we have 2 important lectures. First is current and future treatment and therapeutic strategies against Canadia biofilms, which will be presented by Professor Imbert. And the second is chemical oxidation by Dr. Florence and monofluoramine by Dr. Florence. Chemical oxidation and monofluoramine by Dr. Florence. I believe these topics will give new important knowledge not only for academic but also for industry and government. Therefore, today we also invite participants from industry and government. I hope this intensive discussion will be happening during lectures. Finally, this is a right time for me to officially open this guest lecture, and I wish all of you have useful time of interesting and beneficial lecture. And to honorable speaker, I would like warmly welcome you again. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Saya berharap bahwa acara ini akan sangat bermanfaat bagi kita semua, dan tentu saja hadirin bisa memanfaatkan kesempatan ini dengan sebaik-baiknya. Terima kasih untuk kehadiran para undangan. Wassalamualaikum warahmatullahi wabarakatuh.
Ladies and gentlemen, the second session is a gift souvenir to Prof. Dr. Ayu SHMU, Prof. Imbert Chrition and Dr. Florence. Please. Okay, ladies and gentlemen, the next session is general lecture by Prof. Imbert Chrition, PhD, with topic current and future treatment and therapeutic strategies against Candida biofilms. Continue general lecture by Dr. Florence Berne, topic chemical oxidation by chlorine and monochlorine, chemical and process effect, with our moderator Dr. Hadiana. Please, welcome.
Okay, good morning, ladies and gentlemen. Welcome to the press lecture at graduate school or at School of Postgraduate Studies, Universti, and my name is Hedianto. And today I'm serving as moderator and the chairman of this session. Before we start, I would like to introduce you to honorable speakers today. First is Professor Chrition. Professor Chrition is currently Professor of Parasitology and Medical Ecology at the School of Pharmaceutical University of Poitiers, France. And she's also head of Laboratory of Ecology and Biology of Interactions. And today she will giving lecture on fungal biofilms in human body, how they can affect humans' health. And the second speaker is Dr. Florence. And Dr. Florence is a scientist at Poitiers Institute of Chemistry, Material and Natural Resources, and her specialty is in water chemistry. And today she will give a lecture on chemical oxidation by chlorine and monochloramine, chemical and prospect aspect. We will finish the discussion at 12:30 and then continue by having lunch, and then we can start the first discussion for first lecture by Professor Chrition, and the time is yours now. Thank you.
Thank you very much. I'm stay here. Thank you very much, everybody, to be here. I am very pleased to do this presentation and to show you some main data about biofilms. I am also a teacher of parasitology and medical mycology in pharmaceutical studies school in France. My research topic is on fungal biofilms. So, yes. Okay. So, what is a biofilm? Here's the point. In fact, in natural conditions in the nature, microorganisms doesn't live as planktonic in planktonic conditions. They live in biofilm. They live attached to a surface. So we have to make the difference between these two conditions: planktonic, when microorganisms are not adherent, and when we have sessile microorganisms, so they adhere to a substrate, we can speak about biofilm. This is very important. So a biofilm is a complex structure. We will see later what is in this complex structure exactly, but these pictures are pictures made in my lab about fungal biofilms. And the definition of a biofilm is that a biofilm is a structured consortium of microorganisms that are embedded in a very complex matrix, self-produced polymer matrix. So the matrix is produced by the microorganisms themselves. So where are biofilms? Biofilms are everywhere. They are everywhere around us in our environment. And my colleague Florence Berne will speak about the impact of biofilms in water a little bit. And each time we have a flow, each time we have a liquid, water, serum, saliva, and other liquid, a biofilm may develop. So this picture shows you some example of conditions promoting the formation of biofilm, and of course we have the same in our body, as biofilms can develop everywhere each time we have a liquid. So, for example, the most well-known biofilm is the dental plaque. The dental plaque, which is composed of at least 500 species of microbes. So it is a huge biofilm. You have also some biofilm that form on the surface of contact lenses, in the luminal part of catheters, and I am very interested in this kind of biofilm attached to catheters, and also in other conditions, prosthetic devices, and so on. So finally, this picture summarizes the different conditions where a biofilm can be found in our organism, and it's difficult to read, but if you look on the if you look on the red walls, in fact, you can see all the medical devices that can be substrate for the development of a biofilm. And on the right part, in blue, you can see that a biofilm is able to develop not only attached to a medical device, but directly to the tissue or the cell of our body. So sinusitis, endocarditis, vaginal infections, chronic wounds, etc. are very often involving a biofilm. No? Yes. So a biofilm means two important things. First, microorganisms are adherent to a substrate and with each other, and there is a production of a matrix by the microorganism, and this matrix explains that the biofilm is a very dynamic structure which is always reorganizing. So it has to be considered as a living structure. So sometimes biofilms are including only one species, microbial species, and we speak about single species biofilm. Sometimes they include a lot of different species, and when there are at least two species, we speak about, we can use the word mixed biofilm or complex biofilm. So when do we have single species? When do we have mixed biofilms? In fact, it depends on the conditions. Usually when we have a device such as a vascular catheter or a prosthetic cardiac valve, the biofilm contains only one species. However, when we have some urinary catheter or dental prosthetic materials, etc., we have complex biofilm, including more than one species, and we have some key couples of microorganisms, such as Candida albicans plus Pseudomonas aeruginosa, etc. Actually, biofilms may contain only fungal species, or only bacterial species, or both, and mixed in this case fungal and bacterial species. So my topic is especially focusing on fungal biofilms, but I also study biofilms including fungal mixed with bacterial microorganisms. So what's wrong? What's the problem related to biofilms? In fact, biofilm, so I gave you previously the definition of a biofilm. It's a consortium of microorganisms. So it is a reservoir, an infectious reservoir. And the problem is that some of the microorganisms in the biofilm can detach, can be removed, and they will be free, and once they are free, they can be responsible of the infection. So if we consider this way, the biofilm is not directly the infection, it may be responsible for the infection. So in this case, biofilm is a reservoir of infection. But there are some other types of biofilm that are directly infections. And this, for example, this type of biofilm is, I can give some example such as chronic wounds or biofilms in respiratory tract, in cystic fibrosis patients or other chronic pulmonary diseases. So in these conditions, the biofilm is directly the infection. It's not only the reservoir, but it is the infection. So, in fact, another important point is that where the biofilm is developing is associated with the failure of the local immune defenses of the host. So this failure in local immune defenses is really an issue and is explaining why we have chronic infections associated to biofilms. So literature data say that biofilms, including all types of species, are associated, would be associated with 90% of catheter-related infections. So it's huge. And the mortality rates associated to, for example, device-associated candida infections, so fungal infections due to candida, the mortality rates are more than 30%, so very high mortality. And this is the reason why we have to perform our understanding of biofilm to be able to fight them. Okay. Yes. So I studied fungal biofilms, and just to show you the main species of fungi that are involved in biofilms, the main one is Candida species. Candida is a yeast, and there are different species of Candida, and these are associated with the highest mortality, and especially in the context of catheter, vascular catheter. So I am really interested in these Candida biofilms. However, I will also speak about Aspergillus fumigatus and other Aspergillus biofilms, and this is in the context of respiratory tract infections. Cryptococcus neoformans, which is also a yeast, is able to form biofilm in some cases, and also Fusarium and a lot of other species, but they are not so much involved in infectious diseases. So, biofilms, we have two different types of biofilms, and I will illustrate each type, giving you some example. So the first in the first case we have In the first case we have a substrate, for example the catheter or the dental plate, the dental process. So the medical device and some microbial cells adhere to this substrate and form a biofilm. So this is a surface-attached biofilm. And in the second case, you can see that the microbial cells adhere to each other, but not to a substrate. There is no real surface. And in fact, they are attached thanks to host material, such as mucus, for example, in the respiratory tract. And this is the second type of biofilm, and I will illustrate this type using aspergillus biofilm, for example. So let's move to the first case, when we have a surface that can be a linear surface, such as a medical device, but also that can be a tissue, like vaginal cavity, oral cavity, etc. So the main example, the most important in terms of mortality is Candida biofilm. So Candida, different species of Candida can form biofilms, and you can see here some pictures showing biofilms. These are, sorry for that. These are electronic pictures, electronic scanning pictures, which is a very useful tool to study biofilms. And here, the confocal scanning microscopy. The electronic help to see the matrix. Confocal does not. So when we look on biofilms made by Candida, we are thinking about patients with vascular catheters. This is the main context. So how a biofilm is forming for Candida albicans? Candida albicans is a dimorphic, a polymorphic fungal species, so we can find it as yeast form. We call it blastospore, and this is this type. This is the blastospore, the yeast form, and also we can find it as hyphae like that. And both forms are involved in the formation of the biofilm. Both are very important to have a big, beautiful and strong biofilm. So this slide shows you the different steps in the formation of the biofilm. As you can see, first the surface, that it is the line, the black line, is coated by some host components that can be serum components, saliva components, etc., depending where is the device. Then some blastospores, some yeast forms, adhere to the substrate coated by human components and form microcolonies that will become macrocolonies. This is the colonization step. This is the adhesion step. After this first step, we have the maturation phase. And during this maturation phase, you can see that blastospores form hyphae, and also these microbial cells produce matrix. And once the biofilm is organized like that, so this is he adopts a three-dimensional structure, organization, architecture. We can speak about mature biofilm. And it is not drawn on this slide, but after this maturation phase, we have another, the last step, which is the dispersion. And this dispersion is very important because some microbial cells are detaching, removed from the biofilm and become free and are responsible for the infection or for the formation of another biofilm in another site. So finally, if you can see that it is a bilayer organization. If I come back, you can see the anchorage layer and upper another layer. So this is a bilayer organization with blastospores and hyphae above. This is a very heterogeneous structure also. So this is just to show you, but it's not very clear on the screen, the biofilm showing the matrix and hyphae and blastospores. So the problem why we are interested in this biofilm, we are interested because they are responsible for infections, and so we want to find new solutions to fight this biofilm. The main problem is that cells in biofilms are poorly susceptible to treatment. So microorganisms growing as a biofilm are resistant or not susceptible to antifungal agents or antibiotic agents. And the main reason is the presence of the matrix that is called here PCM. So the extracellular matrix is responsible for the resistance. The other factors responsible for the resistance are efflux pumps that prevent the entrance of the state of therapeutic molecules into the cells, and the presence of cells that we call persisters. And I would like to show you what is a persistor. So, persisters are not regular cells. Persisters are tolerant cells. I mean cells that are tolerant to antimicrobial agents. And tolerance is not tolerance is a reversible phenomenon. It's we have to make the difference between tolerance and mutant. So this is important. You can see here on this picture the device, the medical device, the surface coated by a biofilm. In this biofilm you have a lot of regular cells in red and a couple of persisters in blue. So they are not numerous. And these are persisters are not susceptible to immune system, and they are not